All right, well, I'll get started with the introductions, which people are probably less interested in than hearing from our speaker. But on behalf of the Harvard Club of Southern California, I want to thank you for joining us at this Harvard Speak LA virtual event. The Harvard Club of Southern California is one of the largest and most active Harvard alumni clubs in the world, and we're delighted to be able to sponsor this event. My name is Neil Morgenbesser, and I am Harvard College class of '86 and the director of the Harvard Speak LA program, which brings the opportunity to the broader alumni community for some intellectually and academically stimulating interactions with some of the most renowned Harvard faculty members. Our 2025 talks have so far included Dr. Loretta Mickley on wildfires, astronomy professor Avi Loeb on evidence of extraterrestrial intelligence, law school professor Randall Kennedy on his upcoming book on the demise of Jim Crow, retired government professor Harvey Mansfield on conservatives at Harvard, philosophy professor Ned Hall on how to build a culture of intellectual vitality at Harvard, and government and sociology professor Theda Skocpol on America's democracy and governing crisis. Today, we're extremely excited to be able to hear from Professor Joel Hirschhorn. Dr. Hirschhorn is Chief of Endocrinology and the Concordia Professor of Pediatrics and Professor of Genetics at Boston Children's Hospital and Harvard Medical School, and an Institute member of the Broad Institute. His research focuses on genetics of common human diseases and traits, especially those related to body size like height and weight. He chairs the GIANT Consortium, which has discovered many of the genetic variants known to affect obesity and height. His lab uses largely computational approaches to study how these genetic discoveries can lead to new opportunities for therapy for obesity or growth disorders, and how genetic information could help identify children who have undiagnosed diseases that impair growth. Dr. Hirschhorn received his AB summa cum laude in biochemistry from Harvard College in 1986, and his MD PhD in genetics with Fred Winston from Harvard Medical School in 1995. He completed a pediatric residency and pediatric endocrinology fellowship at Boston Children's Hospital and postdoctoral training with Eric Lander at the Whitehead MIT Center for Genome Research. He has co-directed the metabolism program at the Broad Institute since its inception in 2004, and he has helped direct the human genetics course for first-year Harvard medical students since 2006. Dr. Hirschhorn has received multiple awards, including the Society for Pediatric Research E. Mead Johnson Award. He recently served as the president of the Society for Pediatric Research, and he is an elected member of the National Academy of Medicine. Professor Hirschhorn will discuss how genes affect our height, weight, and other things we care about. Over to you, Dr. Hirschhorn.
All right. Thank you, Neil. Thank you so much for the invitation. Thank you for coming to hear about genes, height, and weight, and some other things. As Neil said, I'm also, not coincidentally, class of '86 from Harvard, and look back fondly at my time there and subsequent training. And I'm happy to discuss this with you, and I'll try to leave some time for questions and further discussion as we go. Neil, I'm not sure if you're going to monitor for like hand raises or chats or something like that, but feel free to.
Yeah, I mean, it's up to you, but at the end, we typically have people submit questions in the chat. And if you want to do hand raising, we can do that as well.
If there's something, burning or pressing, I'm happy to be interrupted. And I'm also happy to take chat questions at the end. So. All right, so as Neil said, I'm going to talk about how genes affect our height, weight, and other things we care about. And I'm going to start by doing kind of a little genetics 101 and a little bit of why I actually chose to study human genetics and use human genetics as an approach to understanding human disease and biology. And there's really two reasons, I think, to study human genetics. The first one that I'm going to spend most of the time talking about is that I think that human genetics gives us vital clues as to what are the underlying biological causes for human disease. So we can name disease, we can describe them, diabetes, asthma, cancer, but until we really understand what is the underlying cause, it makes it very difficult to design the best possible treatments for those diseases. And genetics gives clues, as I'll hopefully convince you of, as to what the underlying biological causes might be. There's also a potential more immediate impact in that genetics can provide a diagnosis or provide predictive information to patients, and I'll talk a little bit about that at the end. But for most of this, I'm going to talk about the potential long-term impact of studying genetics. So this is sort of just a schema of what a lot of biological and biomedical scientists do. They're interested in studying some sort of human disease, and they have a lot of different approaches to do that. And the goal of that is really to gain some sort of new biological insight. Eventually, that should lead to therapeutic leads. So clues that, oh yeah, if you attack this gene or this molecule in that cell, that might actually be a good treatment for the disease, and then take those back into patients. So the approach that I take and that my lab takes is to use human genetics as the way to try to obtain biological insights. And there's a number of reasons why I think human genetics is a great approach here. And one of them is that you can do a lot of epidemiology of studying what's associated with a disease. But if you have a genetic association, it's unique because your genes come before anything else. So if you've done the genetic study properly, if you have an association between a genetic variant and the risk of a disease, you know that that variant is somehow causally involved in the disease. So here's a little cartoon about the difference in causation and correlation. And if you find the cartoon funny, then you've understood the difference. I'll let people. All right, we'll go on. So let's see. I have now. There we go. All right. So again, why genetics? Why would genetics have anything to do with diseases? You might say, well, for certain rare diseases, of course it does. Something like Tay-Sachs disease, where you have a single mutation and it causes a disease just in and of itself. We think of those as genetic disorders. But in fact, most of the things that bring people to hospitals and bring people to clinics are also genetic disorders. It's just a different type of genetics. So how do we know that genetics is important for all of those other things? Well, here's a pair of identical twins that I just found this cute picture on the internet. And you can see they kind of look alike. They have the same height. They probably have about the same weight. And if you were to follow them for their lifetime, they would have pretty similar risks of getting a wide range of diseases. So if one got type 2 diabetes, the other one would be pretty likely to get type 2 diabetes as well. On the other hand, if you take people who are unrelated to each other, so this is the cast of one of the X-Men movies, and you can see there's a much wider range, both in physical appearance that you can see, but also in their risk of disease will be much more varied from each other than the identical twins are. And so that distinction of more genetic variability, more phenotypic variability, so phenotype is kind of what we can measure, whether somebody has a disease or their height or their weight. That tells you that genetics actually plays a role in determining whether people are going to come down with a particular illness or not, as well as in their height and their weight. So before I kind of go further, I'm just going to give you a little bit of background into genetic variation in humans. So the first thing you can ask is if you compare two human genomes to each other, so those could be the two genomes I have, one from my mom and one from my dad, or it could be my genome and Neil's genome, or my genome and kind of anybody across the world. And you said, let's line them up and count the differences. How many of the bases of the 3 billion bases in that genome, how many of them are different? So this is a couple thousand bases from one of our chromosomes. And if you looked for differences, typically what you might find is something like this. There's 2 places out of the 2,000. So about 1 in 1,000 bases is different. So we're 99.9% identical to each other, 0.1% different, more or less. So then you can say, well, if I take those differences that I discover by comparing any 2 people, and then I look for them in other people. So, we compare me and Neil, and then we look at the, rest of the people who are on the Zoom call. Do we see those differences again? So let's say if I had an A and Neil had a C, do we see both A's and C's? And the answer is usually yes, we do. And the reason for that is that most human genetic variation that you find when you compare two genomes is actually common. It's widely shared, often widely shared across multiple continental ancestries. And so that if you find a genetic difference, you'll find it in lots of other people all around the world. All right, so a lot of it's common. And where is it across the genome? So it turns out that variation is spread pretty evenly across the genome. As I said, there's 3 billion bases. And you might think that the differences that affect height or weight or susceptibility to disease are going to be in the parts that code for the proteins. So parts of the genome are in the genes themselves, and in particularly what's called the exons of the genes. Those are the parts that determine the amino acid sequences of proteins. And the coding region, the protein coding region of the genome is about 30 million bases. So it's about 1% of the genome. So what that means is that most genetic differences are actually common and non-coding. So you might say, well, probably the non-coding ones, those are not going to be the important ones. But you can then ask, does evolution tell us about whether the non-coding regions are important? And you might do an experiment where you would say, let's compare the genome of a human to the genome of a fish. In this case, the puffer fish, which was sequenced early for some reason. And if you did that comparison, pretty distantly related, you would actually find that most of the things evolution has cared about, that is sort of kept similar between the fish and the human, are actually the regions that code for the proteins. But if you went a little bit closer, so now human and mouse, so two mammals, it turns out that the majority of the regions that evolution has cared about are actually non-coding. And so these don't determine the amino acid sequence. They might determine, like, if you have a gene, not which amino acids are in it, but when that gene is turned on or turned off, or what tissues it's turned on or turned off in. So that suggests that the non-coding variants are going to be important as well. All right, so the model that we have is that there are these, 1.1% of the genome is different between any two people. Probably most of those base changes don't have any effect on height or weight or diabetes risk or anything like that, but some of them do. I'll show you that most of the differences that we discover, particularly when studying common variants, are actually typically modest effects. So it might be a millimeter of height, or a 1% increase in your risk of type 2 diabetes. And so most of the differences then that we see in this picture are actually due to the combined effects of lots of variants in lots of genes. And that's something that's called polygenic inheritance. So that's very different than the way that, for example, somebody would get Tay-Sachs disease. This is instead the reason that one person is taller than another is due in significant part to the combined effects of lots of variants of very small effect. That's not always true, though. So, for example, in this X-Men cast, this is Peter Dinklage, who is a great actor. And he actually has a single base change, I'm pretty sure, that has an effect on height of about 16 inches. So although most of the variants that we study have very small effects, sometimes you see variants with very big effects. And that's sort of like the equivalent of the variants that cause what are called single gene disorders, or monogenic, as opposed to the polygenic variants that I just told you about. Okay, so I showed you this slide, and our goal is to use human genetics then to find the variants that affect things like height or weight or disease susceptibility. So this is that first step in this circle. And it turns out that the approach that you take, either in the lab or on the computer, depends on the type of variant that you're looking at. So if you're looking for the variants that have big effects and cause these single gene disorders or monogenic disorders, you actually need to look for very rare variants, because typically the variants that cause those sorts of diseases are actually not the types of common variants that I showed you. They're sort of new variants that have arisen recently. And so you actually have to find them in each person that you're studying. So what you need to do is you actually need to sequence, if you're looking for coding variants, what's called the exome, or non-coding variants, the entire genome. But if you're looking for those common variants that are most of the genetic differences that you find, you actually use a different approach called a genome-wide association study. And I'm going to tell you a fair bit about our work using that approach. Genome-wide association studies are sometimes abbreviated to GWAS. Okay, so what is a GWAS? So I'll give you the example of height, which is an example I'm going to use a lot through this talk. And so what you do is you take a bunch of people, you get their DNA, you analyze it, particularly at these places that you already know have differences among lots of people in genomes, so these sites of common variation. You then measure all their heights, and essentially you just look to see, does what they have, right here or right here, like let's say that some people have an A and some people have a C. So you could have 3 different what are called genotypes. You could have 2 A's, you could have 1 A and 1 C, or you could have 2 C's. And you say, do the people who have 2 A's differ from the height of the people who have 2 C's or 1 of each? And so that's depicted here. And you go through and you test all of those places that have been cataloged now as having common genetic variation. So here's one where there's no effect of the genotype. So you say, okay, we've looked at that one. We're not interested anymore. But then you might find something like this, where you say, oh, wow, look, the people with 2 C's are much taller than the people with 2 A's. And this is actually exaggerated. You would never see anything with a common variant with an effect this big. But that's sort of the idea of what you're looking for. So you go through millions of sites in the genome and you test them each one at a time. And then you catalog which ones are the ones that have a difference, that have an impact on height or body mass index or risk of diabetes or whatever it is that you're studying. Okay, so that's been done a lot. And we've done a lot of the ones for height and obesity, but this is something called a GWAS catalog. Each of these little pictures here is a single chromosome in the human genome. And each of the little colored dots is a place in the genome that's been reliably connected to either a quantitative trait like height or body mass index or a disease like diabetes or asthma. And now there are hundreds of thousands of these. And thus far, most of the discoveries have been these common variants. That's partly because GWAS is designed to find those, but partly because that's the way that these traits and diseases work, is that a lot of their genetic influence is from common variants. And I'll show you that direct evidence of that for height. So I think I've told you already that most of these common variants have typically pretty small effects, like a millimeter or less on height. And so you might say, well, why would we even care? Like, is that really a clue that we should care about, that genetics is telling us, or is this kind of almost like, don't bother? What I'll try to convince you, at least briefly, is that even these variants with modest effects can have pretty large impacts on biological discovery. And one of the pieces of evidence was even pretty early on in genome-wide association studies, that if you looked at the variants that were found, and then you looked at the genes that were nearby to those variants, it turned out that a lot of the genes encoded proteins that were the sites of action or the targets for drugs that already existed to treat those diseases. So for example, diabetes, you had a couple, high cholesterol, low bone density, or short stature, or in one case, tall stature. These all already showed up in the genome-wide association study, and there are many, many others that I haven't listed on this slide. So you might say, oh, okay, as a few examples, maybe that's, was true early on, but is there really evidence that genome-wide association studies are pointing you to the right genes to study? And it turns out there's a couple of papers, one from maybe about 10 years ago, and then one a little more recently. And they've systematically studied the genes that are nearby to the variants that genome-wide association studies is finding, and then looking to see, were those genes, or really the proteins made by those genes, were those proteins tried as drug targets? And if they were tried, did they succeed? So they looked at a variety of things, but the main thing that they looked at was, okay, you have a drug, it really does work on that protein, and it really has the effect you're looking for. Did it actually help with the disease that you were trying to treat? And turns out the likelihood of it working was directly related to whether there was genetic evidence from human genetics supporting that as a drug target. All right, so I'm going to give you one example where it's gone all the way to completion. This is still sort of the poster child of genome-wide association study, and I was lucky enough to be somewhat involved in this story towards the beginning. So sickle cell disease is a really serious, debilitating disease that can cause anemia, not just anemia, what are called these terrible pain crises, where patients just have bone pain that is just unbelievably bad. And it turns out that a while ago, people had discovered that there was a different type of hemoglobin, not the usual hemoglobin, but the one that you express before you're born, that's in your blood before you're born. The more of that hemoglobin you have, called hemoglobin F, the less bad your sickle cell disease is. So you want to have more hemoglobin F. So we and others did a genome-wide association study of hemoglobin F, both in just people who didn't have sickle cell disease and also in people who did have sickle cell disease. And one of the signals, and this is sort of just the way that people depict the results for genome-wide association studies, one of the signals were these variants right here that were in a gene that nobody had really ever thought about with regard to sickle cell disease before, called BCL11A. And we were able to show that if you had the variant that gave you more hemoglobin F and you had sickle cell disease, your sickle cell disease was actually less severe. So this sort of said, if we could mimic the effect of this variant, maybe we would do something useful for sickle cell. And then fortunately, the person who had kind of approached me with the idea of doing genetics, Vijay Sankaran, who worked with Stuart Orkin, who's both of them amazing scientists. So they had been searching for a long time for what the gene was that might, allow you to make more hemoglobin F. And they showed that in fact, BCL11A was that gene. So when they turned down the level of BCL11A in blood cells, hemoglobin F went up. And then folks in Stuart Orkin's lab went on to show that if you then removed the part of the genome right around that variant, that also made hemoglobin F level go up. And that suggests that if you could genome edit those cells to remove that in people, where actually you take their blood out, edit it, and put it back in, that might work. And in fact, it has. So this is now sort of the first real effective treatment for sickle cell disease. And this is used as a type of genome editing called CRISPR. So this is an example where there was a clue that came directly from genome-wide association studies that nobody suspected. Genetics was basically telling us, this is where you should be focusing your efforts, and that worked. So what we're hoping is to have many more examples like that. So I did tell you that, a lot of these variants have very small effect sizes. So the one in BCL11A had sort of a moderate effect size, I would say, on hemoglobin F levels. But why should things with small effects actually give us those useful clues? And so I think the logic here is that we're studying common genetic variants. And for a variant to become common in the population so that, maybe more than one person on this call would have it, evolution has to have not noticed it. If evolution notices it and it's deleterious in any way to fitness, evolution will make sure that that variant stays rare. So for it to be common, it has to sort of flown under the radar of evolution. And for it to have flown under the radar, that means that the effects on biology have to be pretty small. But it means that you might have bigger effects on the same biology, and that might have more profound effects. So just to illustrate that here, if you have a little change in a gene that slightly changes its function, you might get a slight change in the outcome. So a slight change in height or a slight change in diabetes risk. But if you have a mutation that has a big effect on the gene, like kind of breaks the gene, you might then have a much bigger effect on the outcome. And you could replace that big effect gene with maybe a drug that actually hits the same biological pathway, but hits it much harder than the common variant was going to hit it. And so that's sort of the logic. Now, that only gets you one step of the way, as I'll sort of show you again. There could be many, many steps between the gene, even if you can figure out what the gene is, and then actually understanding what the mechanism is and sort of how you should design that drug. But it at least gives you that first clue. And because genetics is always causal, it actually tells you this is a causal path, and it's one that's worth trying to figure out. All right, so that's kind of the background for genetics and why do genetic studies to try to figure out disease biology. So I'm going to tell you now the applications to height and to obesity that we've been doing as part of the GIANT consortium, stands for Genetic Investigation of Anthropometric Traits. And I wish I could take credit for the acronym, but it was one of my collaborators. And we've basically been organized into a large international consortium that's been working together for about 15 years now to try to discover the variants that influence height and weight. All right, so you might say, height, that's an odd thing to study. So there's a couple reasons I study height. One is that it is really sort of the original classical polygenic trait, and I'll show you that in a couple of slides. And there are also there's a good positive control, essentially. So we know what some of the right answers are. There are these genes, many of them, hundreds of them, that cause monogenic disorders that cause people to be short or have other issues with their, or tall, or have other issues with their skeletal growth. And we call these OMIM genes because they've all been cataloged in an online resource called OMIM. It's also very heritable. Genetics plays a huge role, within a particular population. About 80% of the variation in height is due to things that you inherit. It's also easy and routinely measured, so you can have huge sample sizes, which you'll see in a minute. The other reason I study it is I'm a pediatric endocrinologist, as Neil mentioned, and it's one of the most common reasons why kids and their parents come to see us. And in fact, when I was a first-year fellow training, I would see a lot of kids with short stature, and I would say, well, the reason that your child is growing more slowly than their peers is because the two of you are on the shorter side, and you've passed down some number of genes, and you've passed down sort of the shorter versions of those. We don't know what those genes are, but ... And about the 20th time I said, we don't know what those genes are, I was like, actually, I could figure out what those genes are. And so that's one of the things that I've been doing for the last 15 years. All right. Okay, so this is the sort of, for those who like history of science, this is the very brief history of science through the lens of height and genetics. So some of you may have studied statistics and may have used something called regression. It turns out that regression was invented to understand how height was passed down from parents to children. So Galton, who's the kind of one of the founders of genetics, even before there was a word genetics, he showed that children of very tall parents tended to regress a little bit towards the mean, and same thing for children of very short parents. And that regression is how regression analysis got developed. A lot of statistical tests that have Pearson's name were also invented to explain familiar resemblance between height. Say Galton and Pearson also did some really bad things about, theories of race and eugenics that I'm not going to get into, but just full disclosure there. But they concluded height is strongly familial. Before they were working a little bit, but still unknown to most of the world, Mendel was working in his garden studying peas, and he was studying things that were really turned out to be monogenic. So peas could either be round or wrinkled, yellow or green. The plants could be short or tall, actually. And so these were some of the traits that he studied. And people rediscovered Mendel right at the turn of the century, of the 20th century. And in fact, one of the very first things that was noted to be inherited in people, like the same way the Mendel's peas worked, was a syndrome of short stature and small fingers. So this is actually from a PhD thesis that I'm realizing is actually now overdue at the anthropology library, but was a student at Harvard named Farabee. And this is actually a picture from his thesis. And so he actually traced the inheritance of these people with short hands, short fingers, and short stature, and showed that it was passed down just like a dominant gene in Mendel's peas. And as I said, there are now hundreds of these described. The other way, and very important for what we do, that human height played a role in early genetics is that, so people, reread Mendel, they had found all these things in humans that worked just like Mendel's peas, and said that must be how genetics works. But then there are other people called biometricians who said, well, yeah, you may think that, but when we measure height, it's like a bell curve, right? It's not like there's, a bunch of people who are tall and a bunch of people who are short, and then, a bunch of people in the middle and nothing else. And so this was a huge debate, and every side was calling the other side, completely wrong. And, how could you be so, ignorant? And then a guy named Fisher, said, yes, these are not the same, but in fact, you can reconcile this. And so the idea he came up with was a polygenic trait. She said, yes, there are lots of things that behave just like Mendel's peas, but instead of making you much taller or much shorter, each one of them only makes you a little taller or a little shorter, and then lots of them add up. And when you add up lots of things with small effects, this is like a fundamental theorem of statistics, you actually get a normal curve, you get a bell curve. And so he said, that's how it works. It's a polygenic trait. And he turned out to be right in large part. And we've kind of now proven that he was right. All right. So this is kind of what the distribution of height sort of looks like. There's kind of a bell curve part. There's actually a little bit of a tail here for people who are a bit shorter than you might expect if it was perfectly normally distributed. Most of the bell curve is influenced heavily by polygenic variants, and I'll prove that to you shortly. In fact, in the next couple of slides. So we've been doing genome-wide association studies of height now for 15 years. And our most recent one, we actually got data from about 5 million different people, still mostly of European ancestry, but some now sort of other ancestries. And the upshot, and this is sort of the group of people who are leading this, and particularly Loic Yengo, who's an amazing scientist from Australia. And what we discovered is that we could identify about 12,000 places in the genome that explain really most of the variation height that we expect to find from common variation. And how we know what we expect to find is quite complicated. I'm not going to go into it, but we found these 12,000 things. They actually are not spread across the entire genome. They're concentrated in about a fifth of the genome. So they're really non-randomly clustered next to each other. And they tend to be near these OMIM genes, the ones that cause the monogenic syndromes of skeletal growth as well. We then tried to see how much, if we take these variants and we try to use them to predict somebody's height, we can explain about 40% of somebody's height if they have European ancestry, less if they have non-European ancestry. And that's because our study was so heavily weighted towards people with European ancestry, because that's who we had data from. So we explained about 40 or 45% of the variation, and it turns out that's almost all the common variant, the inherited part that's due to common variants. And if we look sort of right nearby through some complicated methods, we've actually mapped pretty much all of the genome that has that has common variants that influence height, both in European and non-European ancestries. We've done similar things trying to look at measures of obesity. In particular, our lab has been looking at body mass index. And one of the things that we found relatively early on, and has stayed true as we've increased our number of people that we're studying, is that the genetics clearly implicates genes that are expressed in the nervous system. And what this tells us is that obesity is really, you can think of it as a neurobehavioral disorder. And some of the genes were quite familiar. There's a pathway that had been well worked out from studies of the unusual people who had monogenic disorders of obesity, also from studies of mice, and those are some of these genes that are here. We also did find quite early on GIPR, which turns out to be now one of the two targets of a drug called tirzepatide, which is one of the most effective therapies. It's a type of GLP-1 agonist that I'm sure you've all heard about, with Ozempic, Wegovy, et cetera. And GIPR, if you add that on, it actually makes the GLP-1 agonists more effective. So, but there were also lots of genes who had like no clear connection at all to body weight regulation. And so what our hope is that those genes are giving us clues to potential new and maybe even better therapies. All right, so we've done this genome-wide association study. We also got about 10,000 signals, although unlike for height, we haven't yet mapped all the regions that have common variation. BMI genes are spread a little bit more over the genome. All right, so I showed you this slide. You know, the idea is that you get new genes that are implicated by genetic studies. Those should be new therapeutic leads. But I also told you that a lot of the variation that we find in genome-wide association studies is non-coding variation. And so that gives us a little bit of a problem in this kind of next step that we'd like to take. So we've sort of done that first step. We'd now like to turn those variants into biological clues. But because it's non-coding variants, it actually turns out to be hard to know which variant is, which gene is being affected by the variant that we've discovered, and for some other reasons that I won't go into as well. And even if you could know the gene, there's actually lots of steps that you need to take to figure out what is the biology, how can you actually target the biology that nature has left us a clue about. And there's a variety of approaches that we and many others take. I'm going to tell you briefly about a couple of them. So the first one is just computational approaches. Our lab is a computational lab, so that's one of the things we like to do. And so when we got the first GWAS for body mass index, and we had all these genes that we couldn't recognize, we tried all the methods that were out there to try to predict which genes were relevant, and none of them worked. So we actually had to invent a new method, and it was called DEPICT, and it was by Tuna Pears, who was in the lab, and colleagues in Groningen. And I'm not going to tell you about the details of how it works, but it can point to both what tissue or what cells the genes are likely working in, what sort of biology they are representing, and even help prioritize, which genes are likely to be the right ones. So when we do that for height, we get the right cells, or at least some of the right cells. So chondrocytes are cells that make cartilage, and they can live inside in children in a place in your bones called the growth plate. And the way you grow is that these chondrocytes actually divide and then get bigger and then make bone and push the ends of your bones apart. And that's basically how you grow. And then mesenchymal stem cells are actually the cells that chondrocytes come from. When you try to look at the biology, it gives you sort of relevant things like skeletal system and bone morphology and things like that. And then when we tried to predict which genes might be relevant, we actually got the second one on the list was this one, that right after we put it on our list, it actually got published as causing a skeletal growth disorder. So we're like, okay, this seems to be working pretty well, but seems to be working pretty well is like not really very rigorous. And, sort of one-off, maybe we got lucky. So we wanted to know, like, are we or other people who develop methods actually identifying the right genes or the right, biology, the right sets of genes? And the problem is that we don't know what the answer is. So we have a hard time judging how well we're doing. Like we can, I can run a computational program and it can spit out a list of genes and I can assert that those are the right genes, but how can I prove that? So with no gold standard, we had to come up with a different method, and a graduate student, Rebecca Fine in the lab, came up with something called benchmarker. Again, I'm not going to tell you how it works, but trust me that it is actually unbiased and data-driven. And so we actually then used that to compare 3 different methods, that one that we had developed, one that we helped develop, and one that we didn't develop, and compare them. And it turned out that they didn't do all equally well, but the one that did the thing that did the best was actually the genes that were in the intersection here, so that had been pointed out or implicated by all 3 methods. And so using this metric that Rebecca developed, the genes that were in this intersection of these 3 methods called POPS, MAGMA, and DEPICT, those actually did the best. So we now have a list of 356 genes, and one way of at least doing a sanity check is that we look to say, are any of those these OMIM genes that cause the monogenic disorders? And it turns out that 16% of them are actually OMIM genes, as opposed to, say, 3% of the genome is OMIM genes. So there's a high enrichment. So we think these are likely to be very plausible genes that as for influencing skeletal growth. You can also do more genetics. So this is kind of the other thing that our lab likes to do. I'm skipping the experimental stuff that we do. And so I showed you early on, if you want to study common variants, including non-coding variants, you have to use a genome-wide association study. Turns out there was a special kind of genome-wide association study that could focus on coding variants, including ones that were a little bit less common. And so we did that. And I'm not going to take you through all of this pathway here, but we identified a gene called STC2. And if you look at this pathway long enough, you'll predict that if you decrease STC2 function, you should increase height. And the 2 variants that we found increased height, and a collaborator class Oxfig showed that in fact they decreased STC2 function. So if you could inhibit or block the effect of STC2, that should actually increase height. Well, what about really rare variants that are out of reach of GWAS studies that you have to do sequencing to find? So there's now been a fair bit of whole exome sequencing, not by us, but by people sequencing from large biobanks like the UK Biobank, and more recently the All of Us study from the United States, and a whole bunch of others. So I'm going to tell you briefly what they found. So if they do, when they've done whole exome sequencing, what they see is these are the sort of the best genes. So this is like the genes on chromosome 1 all the way through the genes on chromosome 22 and X and Y. And these dots here, the ones that are high up that look like little airplanes above a city, those are the ones with the best evidence from exome sequencing. So there's good evidence that having a variant in this gene that changes the protein coding sequence affects height. And a lot of these are very familiar genes. So like aggrecan is both an OMIM gene and a gene that was prioritized both by those 3 different methods. And you can see a lot of the other genes as well. And in fact, they are enriched not only among the genes that are, survive the statistical penalty that you need to apply because you've tested 20,000 genes, But even the ones that didn't survive that, but are have a p-value of 0.05. So, expect by sort of random chance, at least 1 in 20 genes to have a p-value of 0.05. But it turns out that the ones that are even just a little bit beyond that threshold are still enriched. So there's a lot of clues that are being given by this exome sequencing as well. And so what we're hoping is that as the sample size for the exome gets bigger, we can cross-reference that with our genome-wide association study and our computational prioritization, and we'll get lists of genes that are really, really robust as being height genes. Okay, so I'm going to summarize what I told you about the long-term impact of genetics. You do genome-wide association studies, you get many variants per phenotype if you have enough people that you're studying. Most of the variants thus far are non-coding with small effects, but you can still have a lot of biological importance even for small effect sizes. But it's hard to get at what the right biology or what the right gene is. And there are lots of approaches that people are trying. And this is kind of the bleeding edge of the field, is how do you get from GWAS results to the genes that you should be focusing on for therapy. All right, so for the last little bit of the talk, I'm going to tell you about sort of more immediate impacts. So one slide just on monogenic disease diagnosis. So this is kind of in some ways obvious, like if somebody has a disorder and you sequence their genes and you find the mutation, that actually has an immediate clinical impact. And this is that distribution that I showed you before for height. And there's a tail here, and part of the reason there's a tail here is that some of those folks have these monogenic disorders of short stature. Turns out that depending on how you collect your patients, about 10 to 40% of children with severe short stature, so like 3 standard deviations below the mean, have a monogenic disorder. But usually unsuspected, or often unsuspected. It's very hard to tell them apart from the kids who don't have a monogenic disorder. So sequencing is potentially quite beneficial there. But could polygenic scores, so polygenic variation, have an immediate clinical impact? So that's something called a polygenic risk score. So I'm going to tell you about that briefly. So this is how it works. So you might have a couple of different people here, the 12,000 variants that we identified as being associated with height. And you can write down for each of them, what is the effect on height? So they might be like fractions of a millimeter. And then you can count for person number 1, how many at each of these variants, how many height increasing alleles do they have? And then you can multiply, or subtract, depending on whether it's 2, 1, or 0, the effect of height. So, in this case, they have no height increasing alleles, so you would subtract a little bit of height. In this case, they have 2, so you would add a little bit of height. And then you sum that over all of the 12,000 variants, and you say, oh, this person should be a little bit taller than average, whereas this person who has different genotypes should be a little bit shorter than average. And that's kind of the way polygenic scores work. And you can do that for quantitative traits like height, or for diseases like heart disease or anything else. So in fact, people have done that for heart disease, and this is actually now many years old, so the prediction power has only gotten better. But even in 2018, what say Katharesan and his colleagues showed was that you could identify people who were in the top, say, 2.5% of a polygenic score for a heart attack or myocardial infarction, and they actually had a risk that was equivalent to people who carry a monogenic variant called in the LDL receptor, so familial hypercholesterolemia, that we routinely use to treat that. If you find that, you treat them with lipid-lowering drugs. And so these folks should also probably be routinely treated with lipid-lowering drugs. Skip this slide. But basically, I showed you this, that there's a number of kids who have monogenic disorders of short stature, and we often have a hard time telling them apart from the kids who are just short from polygenic variation. And so this leads to the hypothesis that if you measured the polygenic variation in these children, maybe that could tell you who's more likely to have the monogenic disorder. So we're actually testing this, and the data have just hit the lab, but we haven't analyzed it yet, so I can't tell you the answer yet. But we have a collaboration to say, if you have a kid who's short, but their polygenic score says, yeah, you should actually be average or maybe a little taller than average, are they more likely to have something else going on? And that something else could be a monogenic disorder, or it could actually be like a disease like celiac disease that is impairing their growth. And so we want to find out, is a polygenic score useful in the clinic? We've also, and the GIANT consortium, and this just came out recently, explored how useful could obesity prediction be in childhood. And so this is, if you look at now the bottom 10%, the middle 80%, or the top 10% for now a polygenic score for body mass index. Then as people age, they kind of start the same, but then as they get older, and this is taking them up to their early 20s, you can see a spread here starting to happen where the people with a higher polygenic score have much higher BMIs than the people with a lower polygenic score. You might say, well, I don't really need to predict BMI. I can just measure it, right? And that's true. And in fact, if you do it in adulthood, the genetics only add very modestly to prediction later in life. But if you do it very early on, it turns out that measuring, young kids' height and weight actually has a little bit of predictive value, but almost none. And if you then add the polygenic score, you actually substantially increase the amount of prediction that you can get of what their BMI will be like at 18 years of age. By the time they hit age 8, most of that value has gone away. All right, so one last example. Of could this sort of genetics be clinically useful? And this is for type 1 diabetes, the other sort of most common thing that I see as a pediatric endocrinologist. And it turns out that most of the genetic risk for type 1 diabetes is already explained by variants that have been discovered. And it's partly because it's an autoimmune disease with a huge effects of a few common variants. So you can make a polygenic score for type 1 diabetes where some people have a very low risk and some people have a very high risk. And I think I'm missing an animation here, but it turns out that the people who are in the top 3% or so have something like a 20-fold increased risk of getting type 1 diabetes compared to the average person. So Ray Krankamp has used that. Oh, no, actually, I'll say that later. But Ray Krankamp, who's a new faculty in our division, has actually used this now in the clinical setting. So he went through and he looked through a biobank that we have at Boston Children's Hospital and categorized all the people with diabetes into their type of diabetes based on their medical record. And so what he saw was that the people with type 1 diabetes had much higher type 1 diabetes polygenic scores. That made sense. And the people with other types of diabetes had much lower scores. And then there was this one person here who had a really high type 1 score, but they had been characterized as having just high blood sugars from medicine they were taking, from steroids. Here it is. And it turns out that this person actually still had high blood sugars even when they went off steroids. They probably actually have type 1 diabetes. And this is just for those who like things called receiver operating curves to show it actually does a pretty good job of predicting what type of diabetes you have. So there's a question you could say, like, would newborn screening for type 1 diabetes risk make sense? So obviously, once somebody has diabetes, they have diabetes, but, should we or could we screen for this as a newborn? And why would you even? So here's the statistic that I quoted you. And it might be that, the people at the high risk would be more on alert for the symptoms of diabetes, and maybe their child wouldn't get quite as sick when they were first diagnosed, because about a third of people end up very sick with diabetic ketoacidosis. But you'd have to trade that off against anxiety of wondering whether my child's going to get diabetes. But it could actually set the stage for prevention of type 1 diabetes. And there's actually a pilot in North Carolina trying to do newborn screening and kind of seeing how that works. But the prevention is now starting to be possible. So there's a drug called teplizumab that if you give it to people who almost have sort of full-blown type 1 diabetes, but don't quite, but very high risk, almost certain to get it, it turns out that you can at least delay the onset of type 1 diabetes by some years. And this is just the first one of these. So we're hoping that there are going to be better examples of this as well. And so that would mean that you could, if you identified people who are at high risk, right now these folks are identified typically because they have a parent or a sibling who has type 1 diabetes, so they get tested. But if everybody had their genetics, it might be that the people at the very top of that polygenic score should also be followed carefully. And this is now being implemented by a team that includes Ray and a bunch of others from our division. And we're actually currently the only center, or I think still the only center in New England doing this, and a leading center nationally. We're in the process of scaling up, but we could readily envision the role of genetics playing a role in who should actually get this treatment or be screened so they could possibly get this treatment. All right, so the summary of what I've told you for the second part, sequencing can identify people with monogenic diseases, which is often clinically useful, but polygenic scores now already have pretty substantial predictive power. very similar to the predictive power of like measuring your cholesterol or your blood pressure, which we do routinely. But how useful it is will depend on what interventions are available. But we are getting closer to what used to be complete science fiction. So for those of you who've seen the movie Gattaca, it's like, when the baby's born and they do a drop of blood and they say, oh, you have a this % chance of coronary artery disease and this % chance of diabetes. So we're not going to screen out all those people because everybody has risk of disease. But in terms of our ability to predict that and maybe do some sort of preventive measure, we are actually closer to that world than certainly we were when I started my career. I've tried to acknowledge people on the way. I'll just leave this slide up, but lots of people in my lab, lots of people from the giant consortium, particularly these folks here, and be happy to take questions.
All right. Well, thank you very much. That was informative, and I followed some of it, so that's good. Let me start. We've got about half a dozen questions people have already submitted, and if you do have questions, you can put them on the chat thread as well. Let me start with, what do you think the biggest question you've answered so far is? And is there a big question out there you want to answer, or is now a bunch of little, maybe not little, but medium questions is just the application of this to different diseases and conditions?
Yeah, so I mean, I think that we've successfully answered. It would, I think, be that we've defined the role of common genetic variation in a polygenic trait pretty completely. So we've showed sort of what happens when you take this all the way to, pretty much all the way to completion. What do you get? What does it look like? And it kind of helps give people a sense of like that there are, in fact, thousands of genes that are involved, all with sort of small additive effects, just like Fisher predicted, now over 100 years ago. I'd obviously love to be able to take some of these successfully to, here's a gene that was implicated by our GWAS for obesity. That's taken us to a therapeutic lead. I'd love for that to happen. In some ways it kind of did already with GIPR, but it was kind of fortuitous that the pharmaceutical companies got to there. It wasn't from our work, to full disclosure. But the big question that we haven't answered is there are all these non-coding variants, and I speculated as to what they might be doing, but really we have no idea what the vast majority of them are doing. And that's, I think, a big question that just sort of is this building block of human biology and human variation. A lot of it's determined by these common variants, and we really have no idea what they're doing. There's not really great evidence that they are actually turning on genes, at least in ways that, or turning them off in ways that we can measure right now.
Okay. Ellen Liao asks, since this genetic analysis requires massive quantitative analysis, how much AI have you employed to help do this analysis? And what do you think the future implications of using advanced AI to help you in this endeavor?
Yeah, it's a great question. So till now, very little, or certainly in our lab, very little. There are AI methods. There was one that was just put out on as a preprint from the same folks who did DeepMind that tries to predict the effects of individual variants. They're doing it using data sets that I think already have not particularly shown much of a correlation with like what are these non-coding variants doing. So I'm a little skeptical as to how useful that's going to be for our purposes. But if you have the right training data sets, and we're hoping that, as we sort of narrow down on which are the right variants and which are not, that we should be able to train AI models to then look at sequence and all sorts of other data about the genome and say, okay, this is probably the right variant. And again, if we can make connections to say this variant affects that gene over there, 3 genes over, then we can then also make predictions for other variants. But we need a really good, robust training set, I think, to make it work.
Great. Let me ask you to stop sharing your screen.
Oh yeah, sorry, let me do that.
So DT asks, are there any genomic breakthroughs, Ray, spinal deformities that affect height like scoliosis?
So there have been genome-wide association studies with scoliosis. I would have to go back to the literature because I think the last paper on this I read was a number of years ago to tell you exactly whether we've gotten great clues there or not. I think it's probably in a similar place, except further behind, because it's harder to get people with scoliosis than people with height, because everybody has a height. But there are some clues that are coming out from the genome-wide association studies. but we probably just need a lot larger studies. And again, to be a lot better at going from the clues that genome-wide association studies are leaving for us to understanding what the right gene is.
Shannon Christmas asks, how many geneticists are tracing disease-causing genetic variants to founder mutations that arise from specific ancestors within a genealogical time frame? With the massive amount of data compiled by direct-to-consumer genetic test companies like Ancestry DNA and 23andMe, will we see more of this in the future?
Yeah, so in fact, a couple million of our data came from 23andMe because they, unlike Ancestry, had actually collected very rich phenotypic data, not to mention height and weight. So they're already sort of participating in that kind of thing, but there's a specific question here about founder mutations. So a founder mutation is sort of like it arose at some point in somebody, and then every instance of it in today's population is in a descendant of that person. And you often see stuff like that, for example, in, say, the Ashkenazi Jewish population or the Finnish population or Amish or castes within India, where the variant is a lot more frequent in that particular group than in everybody else in the world. So there are definitely folks who are focusing on specific founder populations and finding lots of examples of those. For height, there's certainly, I can think of one off the top of my head in Sardinia, for example, that is much more common there than it is in the rest of the world. There's also some really interesting stuff that people are doing with genealogy, where you don't actually need the pedigree. You don't need to know who was whose parents. All you need to know is that there are ways of inferring who's related to whom. And Decode first pioneered this, but other people have done it, and now more sophisticated ways. So you can even tell like which parent you got your variant from. So there are definitely ways of doing that. For the common variants, they tend not to be founder alleles because they're so old, they actually predate out of Africa. So they arose when we were all in Africa and before humans actually emerged from Africa.
Okay. Kimberly Williams asks, do you think increasing genetic self-knowledge will ultimately help people make better health choices, or will it create new psychological burdens? And do you think we'll ever hit a ceiling on how much DNA alone can tell us about our health?
Oh yeah, great questions. So for number one, I think it's going to depend on different people, that people have done studies on this. And I think on average, people tend to be fairly relieved, and there's not and or not more anxious, I guess, after getting genetic information or even genetic testing that, didn't have anything. And in theory, it should help people make better health choices. I think there are a lot of companies out there that are trying to use genetic information and sell it in ways that do not help people make better health choices. So I think it'll have to be based on good science. But the psychological burdens, I think, is a really important thing, and there are plenty of people studying it. Probably one of the best known is a guy named Robert Green, who studies this for Alzheimer's disease and testing for one of the variants that is a big risk factor, and how does that make people feel when they get that result. And there's definitely a ceiling on how much DNA alone can tell us about our health. So one of the reasons I study height is that most of the variation in height is from genetics, but most things are not like that. So, type 2 diabetes, a lot of the variability is from non-genetic factors, whether that's just random chance or the environment that you're in, or, other other things that we don't understand. So, you can estimate that by what's called the heritability as to what fraction of sort of your risk is due to genetics. And for most common diseases, it's 50% or less.
Okay, a couple more. How quickly can we expect these genetic discoveries to lead to targeted therapies?
Yeah. So it is not as quick. It was if you were watching the announcement of the sort of first sequencing of the human genome, there were some people there who made comments that implied by 10 years ago, we would have already cured all disease. So it's nowhere near that fast. And part of it is that we need to figure out what are the genes involved and what's the biology involved. I think these things are going to take anywhere from a decade to many decades for any given place. On the other hand, there are now a lot of these genetic clues, so the faster ones will come first. And then personalized therapies, there already is personalized therapies for the monogenic ones where you have a single mutation. All you need to do is reverse the effect of that. For the polygenic ones, I think we're decades away.
All right, well, we're almost out of time, so one last question. On the nature versus nurture debate, how important do you think your Harvard undergraduate roommate selection is to your ultimate success in life?
It was absolutely critical.
Me too. I'm glad to hear it. Well, on behalf of the two of us and our other freshman roommates, want to thank you for joining us for Harvard Speak LA. This has been very educational. I know people enjoyed it, and it will be available for the club membership on the website afterwards to review. I know I will do that so I can actually understand some of what was being said again. But thank you so much. And Ellen said, I've always suspected you're smarter than Neil. Now I know.
No comment. Yeah.
Yeah, we all knew. All right. Thank you so much, Joel.
Thank you for the invite.
Everybody for participating, and we'll see you on the next one.
All right. My pleasure. Take care. Bye.
Bye.